Hi everyone, this is Mark. I'm back with the third, and I think the last in this series on basic molecular biology and virology. If people are interested in just interesting topics in genetics and genomics, I can certainly do more. We'll see how this goes. Okay. The main point of this one is different approaches to therapeutics for treating viruses or inhibiting viruses. And just to start, as I didn't say it last time, remember I mentioned that RNA viruses can be positive strand or negative strand, meaning that, and by positive and negative, that's in terms of information content only, in terms of whether the ribosome can read the sequence directly or whether it reads a copy or the complement of the sequence. So when it comes to single-stranded and double-stranded RNA and DNA viruses, none of this has anything to do with their pathogenicity. They're just different mechanisms that the viruses have evolved, or not really chosen, but have evolved as successful strategies to replicate themselves. And any of these combinations could be, there's relatively non-pathogenic benign versions, and there's much more pathogenic versions for some of them. Not all four occur a lot. There's not a lot of single-stranded DNA viruses in us, they're rather common in bacteria, but there are some in animals and humans. Okay, so the coronaviruses are positive strand viruses, positive strand RNA viruses. And of the many, many RNA viruses that are around, like measles and rabies and cold and stuff, the influenza viruses, specifically the real influenza viruses, the things which cause actual flu, as opposed to cold viruses that we sometimes might say, I've got the flu, but it's actually just a cold. Anyway, those are negative strand RNA viruses. So the reason for mentioning that is that they're very, very different. They're evolutionarily really unrelated. So there's no special reason why a drug or a treatment optimized for treating influenza would work against coronavirus, and certainly a vaccine wouldn't cross-react. Okay. The other thing about the viral biology is that I mentioned that some viruses are surrounded by a membrane. So if this is the cell membrane, and this is the inside of the cell, and this is the outside. So inside the cell, the virus is in there, and it's been making lots of copies of itself and of its proteins, and so it's assembling viral particles. And in the case of envelope viruses, these particles make their way to the cell membrane, and the virus may be encoding proteins, which are viral proteins, that make their way to the cell membrane also, and they attach there. And so now you've got a nascent viral particle that's on the cell membrane, possibly involved with viral proteins. And then the virus starts to bud. Like that. So there's the viral particle, and in there's the genome. And then eventually the virus buds off, and this is the virion particle. So for these viruses, which we call enveloped viruses, their outer layer is a lipid layer, and that means it's susceptible to detergents. Because detergents, the same reason detergents work to clean our hands or clean the grease off our hands when we've been cooking or when we've got oil on our hands, is detergents are good at dissolving greasy substances into water. So normally oil and water don't mix, but you add a detergent and you can get them to mix together, and then you can wash it off your hands and down the drain. So for the same reason that detergents do that, they are good for disrupting envelope viruses. And now viruses which are just pure protein, depending on how tough the viral proteins are in the genomes inside, depending on how tough the viral proteins are, it may or may not be susceptible to detergents. Certainly if you treat it with strong acids or bases, or treat it with super strong detergents, they're protein, and so there's a limit to how stable they can be. But those kinds of super strong reactions would be toxic to us too. So you can't swallow some hydrochloric acid because you've got an adenovirus. It would kill the virus, but it would kill you too. So the good thing about envelope viruses is that normally if you disrupt the membrane envelope, that inactivates the virus. So that's the reason why they're recommending people wash their hands with soap for COVID-19. And now soap, even for any virus or anything that was stuck to your hands that you touched and was on your hand, if you wash with soap, it's easier to wash it off. But it's more than just that. The soap actually deactivates the virus. And in fact, it works better than alcohol in the sense that alcohol can also deactivate the virus, but from what I've read, it needs a very high concentration of alcohol, and not all hand sanitizers necessarily have that. That's also bad for your skin, and so it doesn't really, you don't need it from what I've been reading. And now, I would definitely say keep watching for up-to-date information in case something new is discovered, but from what I've read so far, washing your hands with soap is very effective, but you have to give the soap time to work. So you can't just slather soap on your hands for 2 seconds and then rinse it off. You've got to rub your hands thoroughly, and it's very complicated. And there's a whole, you can find videos on the right way to wash your hands, which I never did before. It's not necessary for just getting some cooking oil off your hands, but to really make sure that if a virus landed on the back of your hand, or that you opened a door or picked up something in the store and it's in your hand, there's a viral particle and it's between your fingerprints or in your skin somewhere, you really need to give the soap time to work. But if you wash, and so they came up with the 20-second rule. Obviously, well, the longer the better, up to a point of diminishing returns, I guess. So that's why soap is good. And why hand sanitizers are not automatically better. They might be, but just because somebody says hand sanitizer, that doesn't necessarily mean it's better than soap. It might actually be worse than soap, depending on what's actually in it. So you have to be careful about that. And pretty much, from what I read, pretty much any kind of hand soap can work. Maybe the stronger the better, but any soap that's strong enough to wash your hands in should be good to some extent. Okay, so now when it comes to making antiviral drugs or therapeutics, there's 2 approaches. One is direct drugs that inhibit the virus, and the other is vaccines. So in terms of drugs that may inhibit the virus, as I've said, there's 2 approaches. One is you can try to target a viral protein, a viral enzymatic activity, or you could try to target a cellular process, or you could try to target the attachment process. I guess that would sort of be a combination of the 2. In terms of viral proteins, you really want it to be an enzyme. It's, for the most part, enzymes are easier to inhibit because they have active sites that are normally meant to bind small molecules. So they're easier for drug companies to target. And in the case of COVID, the 2 main targets that people have come up with are the RNA-dependent RNA polymerase, because we don't have such an enzymatic activity functioning, or essentially, in our cells. So if you can come up with something that's specific to that, that's good. And another is the protease, because even though we have lots of proteases in our cells also, the COVID viral genome encodes a protease activity that's necessary for the maturation of other viral proteins. And so that's a potentially specific target. Attachment, now you're talking about a viral protein, but a surface protein on the surface of the virus, and you're trying to block its interaction with the cell membrane. And that could have, in theory, deleterious effects on the normal function of that cellular protein. So that's why it's kind of a merger of these other 2 processes. And cellular processes, again, in general, you don't want to be inhibiting cellular processes because they could be toxic, but it's possible that you could do it in a dose-sensitive way so that, as I suggested in the last video, maybe you could slow down the cell process enough so that the viral reproduction inside your cells is just slowed down to the point where your immune system can work against it, can have time to act on it. And I'm not sure how often that's used as a process in general in virology. So From what I read, the World Health Organization has just launched a very large drug trial called Solidarity, and they're testing a couple of fewer than half a dozen, 3 or 4 different drugs that are already out there. So developing a drug from scratch takes a lot of time, but because COVID, SARS-CoV-2 is an RNA virus, there are other drugs already made against other RNA viruses. And so the simplest thing to start with is to see if any of those drugs that already exist, that we already know how to make and that have already in some cases been tested, at least for safety, if not efficacy in humans, it makes sense to try them right away in case we get lucky and one of them works. So I thought I would just go through, since this might be, you might be seeing this if you're reading enough into the media reports, so I thought I would just name a couple of them and describe what they are. So one is remdesivir. So that VIR on the end of drugs is often used when so the name doesn't really tell you anything about what it does. It's just some name they've made up, and I don't know how they do that. But the VIR on the end is found on a lot of drugs that are antivirals, and it's kind of that's a V and that's an R. That's just for marketing purposes. So it is an inhibitor of the RNA-dependent RNA polymerase, and specifically, it's a base analog. So RNA polymerases have binding sites for binding the A, G, C, and U that they're going to use to make a new strand of RNA. And you can imagine coming up with a molecule that looks like one of these that can get into the active site of the enzyme and inhibit it one way or another. But the thing is that we have other DNA-dependent RNA polymerases, as I said in our last video. So you need to come up with a base analog which looks, for whatever reasons, the viral RNA polymerase is blocked or inhibited by it, but our normal cell RNA polymerases aren't. And this drug was actually developed against SARS virus, if I understand correctly. And it was shown to work in animal models, but I'm not clear if it was ever tested in people. It's possible they never got that far because vaccines came along in time, or the epidemic just wore itself out before they got to it. So my understanding is that it hasn't been, I'm not clear on whether it's been tested in people before. That's an important point because there are many, many examples in the pharmaceutical business of drugs which worked fine in lab, in cell culture, in lab, in human cells, or in animals like mice or rabbits or pigs or whatever. And then when they put it into people, it didn't work for different reasons. Maybe our bodies metabolized it, degraded it too quickly so it couldn't work, or maybe it couldn't get to the right part of the body that it needed to, where the virus was attacking, because not all viruses attack all cells in our body, so there's specificity to the delivery, the bioavailability, or maybe it just doesn't work in our cells for some unclear reason, or maybe it's toxic in us and it wasn't toxic in the mice or the rabbit. So testing in humans is very important. There's another drug that they're testing in this trial is lopinavir. And it targets the viral protease. But it doesn't, because it's not a targeting of coronavirus protease. It actually was a drug developed to target the HIV protease. So it's not at all obvious to me why this should work against the coronavirus protease. But there's some report that it might have. So my guess is that it won't hold up because these are not related viruses at all, particularly just because they're both RNA viruses. But maybe we could get lucky. So, and I don't know the exact mechanism by which it inhibits the protease. But apparently, for whatever reason, in us, it's known that lopinavir is degraded quickly. So it's a chemical molecule, and our bodies are actively surveilling and detoxifying anything that they think is foreign. So another problem with just action of any drug in us, if it's a small molecule, is our body's trying to degrade it because it doesn't realize that it's helpful for us. So apparently there's another chemical, which they call ritonavir, which inhibits the process of breaking down lopinavir. So it stabilizes, it targets the degradation. So it stabilizes the lopinavir. So this is a cocktail that stabilizes the ritonavir, and they're using it with or without interferon beta. So interferons are immunostimulatory molecules, or they're immunomodulatory molecules, and they must have some reason for thinking that this might work together with something that stimulates the immune system. But it's not obvious to me that there's any relationship mechanistically between a protease inhibitor and the interferon. And the last one they're testing, so this is the protease, and the last one that they're testing in this is chloroquine, which is, to my mind, the most dubious. And so chloroquines are drugs that are antimalarials. Malaria is not a virus. It's not even a bacteria. It's a eukaryotic monocellular organism. And it's not obvious at all why chloroquine should work against coronavirus. From my limited reading, I don't know that much about chloroquine, but from my limited reading, it seems to block a number of cellular processes, and it's not very specific to malaria. So maybe there's some reason to think that some of those processes might be coronavirus relevant, engaged. So I'm not sure why it got tested or by anybody in the first place, but now there's been enough media on it that the World Health probably felt that they just had to try it. However, anybody who will go unnamed who might be saying that they feel good about chloroquine, that's gibberish. We have no serious information about it at this point, and I don't see why it should work, but maybe it's in that category of things which could just slow down the cell enough so that our own immune response could take over. But if we don't mount a strong enough immunoresponse without vaccination in advance anyway, it's not going to work. So we'll see. Interesting, these are not double-blind placebo trial. It's not a double-blind placebo trial. I guess they felt like that would just take too long. So from what I read about it, and I'll have, it's possible I could have misunderstood, but my understanding was that they're just going to be giving these, not all of them, but one or another of these drugs to a lot of people. And presumably because the infection is so acute and severe, it'll be fairly obvious if one of these, if any of these is helpful, and we won't need to worry. And the idea that somebody could cure themselves with a placebo effect just because they think they got a drug, that doesn't make a lot of sense for this very pathogenic drug. Okay. Just a few words about vaccines and how we make them and how they work. So our immune system has a lot of different ways to attack foreign objects. It can use antibody molecules, but we also have cells which eat bacteria, eat foreign bacteria, eat cancer cells in us that the cell recognize, that our bodies, if they're capable of recognizing it, as toxic. So obviously cancer cells are being selected to avoid that recognition, and our body's trying to clean them up. So there's what we call a serum response or antibody response and a cellular immune response. Normally when we think of vaccination, we think of making antibodies, and antibodies are protein molecules, and they have a complicated structure. That's sort of a schematic of it. But the idea is that the virus has got proteins on the surface, and if it's an enveloped virus with a membrane, there's still proteins, like flu, there's still proteins embedded in the membrane. And if the antibody molecules bind to the proteins here, they can block the virus so it can't get into the cell. The antibodies are not getting into the virus to attack it from the inside, not normally, I would expect. And the problem is that it takes months for us normally to develop an immune response against a foreign object. So for a pathogen that grows in your body very quickly, the reason we get sick from some viruses and not others is that our immune response is too slow. So the point of vaccination is to prime the body. So there's a step where you basically prime the immune system, prime the system in advance, so that if you get attacked by infected by an actual virus, your immune system is already ready to go because it's already seen it before. But you can't just give people the virus and to raise an immune reaction, because then you get sick. So you have to give them something which looks like the pathogen but is not actually pathogenic. And one way to do that, the original way to do it, was to take the virus and kill it. So you could heat treat the virus or treat it with a detergent or with acid or something that would kill the virus but leave the protein structure sufficiently intact that if you injected the inactivated virus, it would look enough like the real virus that you would get, well, and then we would get an immune reaction against that. And so the body would be primed, and then if you got infected with the real virus, the antibodies and the cells that you had generated against the inactivated virus would cross-react with the active virus. And one problem with this is that if you have to inactivate the virus, so you don't want to leave any living virus in there, but if you treat the virus too harshly, you disrupt the protein structure, and then at the end you make what you what you inject people with, the antibodies that you make wouldn't cross-react with the living virus or the full intact virus. So it's a tricky business to use inactivated viruses because if you treat them too mildly, then you're going to leave a fraction of virions intact, and you can actually make somebody get sick from the inactivated virus, and examples of that are there in the medical history. But now that we have molecular genomics and DNA sequencing and all that stuff, we can take the viral genome and find the sequence of the genes that code the coat proteins. And then we can just express those proteins in a heterogeneous system like bacteria or human cell, some other cell type. And we can overexpress just the viral protein. And in theory, we could inject that into people. And so in theory, this should be completely, it's not a whole virus, and so there's no risk of you getting an infection from this. There's general risks involved with purifying any large amounts of any protein in the lab and injecting it into people. You could have contaminants, or the virus could be toxic, or the protein could be toxic for other reasons. So that's an issue, but that's not specific to this virus. That's an issue for all vaccines. So that's a relatively fast process because we already know something about these viruses. So one question somebody asked me recently was, why are they saying it's going to take 12 to 18 months to make a vaccine against coronavirus when for flu, they seem to be able to generate this season's flu vaccine every year and with only a couple months, and give it to people and have them be protected against that season's flu. So that seems to be fast. So I don't know the answer to that. So this is purely a speculation, and my speculation is that we have so much experience with vaccinating people against flu that we know how to, with each new seasonal flu, we can sequence the genome and find the changes in the cell, in the viral proteins that make it resistant to last year's vaccine, and we can just regenerate, or we can reconstruct the vaccinating molecule, and we know it's going to work because we know how the whole process is working. So that's just a guess, because it doesn't take 12 to 18 months to make a vaccine. As I said, you could just clone the viral genome, overexpress tons of the protein, and just shoot it into people and hope for the best. The problem is you have to vaccinate everyone in the world like they did with smallpox, and that's a massive undertaking. And you can't do it repeatedly because the vaccination itself, at some point, the protein that's in the vaccine is dissolved in something called adjuvant, which is something that stimulates the immune system. And so there's a legitimate reason to think that it's not good to immunize people too many times. So that's got nothing to do with all the mercury and all that stuff that's been proved to be wrong. But you can't just make 20 different vaccines and inject everyone in the world with all of them and hope one of them works. That would just be it's not a question of inefficient. It probably would be toxic. So there has to be a process of figuring out for this particular virus what protein or proteins in the coat might work. Sometimes they don't express the whole protein because if you think about it, if this is the membrane of the virus, and so here's the viral genome inside and there's a protein and there's viral proteins embedded in the membrane, only part of that viral protein is exposed to antibody molecules in the surface. Part of the protein's in the membrane and part of it's facing the inside of the virion. So you don't necessarily want to make the whole protein. You might want to make a fragment of the protein, and that might depend on knowing which part of it's facing out that would be accessible to the antibody. And then you've got to test it in people, and that's just, it takes a while. So if you inject people if you immunize people with a vaccine, you don't find out the next day whether they've made antibodies or not. There's just, the intrinsic biological process takes a while. And so there's a delay there. And then you need to know whether it's actually going to protect them. And that's going to be hard to check here because you can't be taking people that you've given an experimental vaccine and expose them to the virus and hope that it protects them. So I don't even know how they're going to test that. But that's the process that has to be gone through. So it's not that it takes 12 to 18 months to make a vaccine. That's their guess. That's their best estimate based on tremendous experience on how long it takes to develop a vaccine and have evidence that it'll work to the point where you want to mass produce it and inject it into everyone in the world of all ages, from children up to adults. So obviously you want that to be as safe as possible, and you want to have the most effective vaccine before you go mass producing and mass immunizing people. And so there's going to have to be, I imagine they're agonizing over a trade-off of how effective does it have to be and how long do they wait to see if it's effective before they go ahead and mass produce it and mass immunize against it. So I think that's it for this presentation. Thanks for listening. And if people are interested in more molecular biology, I'm happy to do some other interesting topics that are interesting to me. Let me know. Thank you.